So this is part one, and the very first thing you have to do for cell culture is you have to make sure your hood is set up. Okay? And the first thing you have to do to make sure your hood is set up is you need to go ahead and put some ethanol on your gloves and just wipe them down. So now I can put my hands in the hood and touch things in the hood because we want to keep everything in the hood as clean as possible. So in the hood you're going to need cell culture dishes, 5 and 10 ml pipettes, holders for large and small tubes, a pipette man rack, small pipette tips, a marker, the gas jet. In this can there are glass pipettes. We'll need those. And this apparatus here is an aspirator that we will use to aspirate the media off the plate. The other thing you need is a pipetter, and this pipetter is always plugged in when we're not using it. So I'm going to unplug it so that we can use it. And I'm also going to turn on the pump that allows this aspirator to work. So before you can actually turn on the flame, I want you to clean the hood, and we'll clean the hood with ethanol. Make sure to wipe everything down well. And then the next step will be to turn on the gas. What I'm going to do is light a match. And I've kept the light off so that you'll be able to see the flame. You probably can't see that in the video, but the flame is on. I'm going to turn it down a little bit. It doesn't need to be quite so high. And make sure that that's not going to interfere with anything. Now I can turn on the fan and the light, and our hood is ready to go. Before you can actually do any cell splitting, you make sure that your media is warmed, and that your trypsin is warmed, and that your media without any FBS is also warmed. So we're going to get those out of the refrigerator. So what you'll need is DMEM and trypsin and CDMEM, which is complete media that has not only the media but also FBS and an antibiotic and antimycotic. And I'm going to take these to the 37-degree water bath, and we're going to put them in there. Okay, so the water bath is on. We're going to place the CDMEM in the water bath along with some DMEM and trypsin. I want to make sure that the bottle doesn't fall over. And I'm going to replace the top. So in the next step, we need to get our cells, take them to the microscope to look at them and evaluate them before we actually put them in the hood to split them. Before you open this door, which is the incubator for our cells, you want to go ahead and wipe down your hands again with ethanol to make sure that they're semi-sterile. It has two doors, an outer door and an inner door. Now open the inner door. We don't want to keep it open very long. I'm going to get my cells. And then I'm going to take this over to the inverted microscope. So this is the inverted microscope that we want to look at our cells with. I've already taken the cover off. You need to turn it on with this little green toggle switch right there. You should see the light come up. We're going to put the cells on the stage right there. This will limit the light that's coming through, and this is the focus knob. The lenses are underneath the stage. It's an inverted microscope, so you still want to start at 4x and look through the microscope, look at our cells, and evaluate them, but you want to make sure that you go up in magnification all the way to 40x, and make sure that the cells look plump and healthy instead of like little raisins floating around. If they're little raisins floating around, those are dead cells. So now what we want to do, these look good, we want to go ahead and split them. So now what we're going to do is we're going to take our cells back to the hood. So we have our cells and I put them in the hood, but before I can do my split, I need to make sure I get my media, my DMEM, and my trypsin. They're in the incubator over here. And before you can put anything in the hood, you have to spray it down with some ethanol. So I just sprayed it down a little bit, and now I'm going to wipe it off with a paper towel. Put that in the hood. This is my trypsin. DMEM, media without any FBS in it. And then 2 more tubes of DMEM. Just in case we need them. So now we have everything in the hood that we need. So now what I'm going to do is go ahead and get in the hood. Don't sit with your body in the hood, just your arms. First thing you need to do is go ahead and open up your media. Yeah. Get rid of the parafilm coverings on the tubes that you're going to be wanting to use. I'm going to loosen the top of this just a little, of the DMEM and the trypsin. Those are the 2 I'm going to need first. Okay, so the first thing I have to do is I have to get the media that's on these cells, which are growing on the plate, so they're adhering to the plate. I need to get that media out. The way I want to do that is use the aspirator. So I'm going to get a glass pipette. Now you can touch this, but try not to touch the end. And then what I'm going to do is I'm going to put a sterile pipette tip on this glass tube, just by touching it to the tip. Now when you open up the dish, you want to put 3 fingers underneath the dish and use your thumb and forefinger to open it up the top. And you don't want to take the top all the way off and lay it off to the side, because you don't want to break the sterility in the top. So I'm just going to be touching the media. I'm not touching any of the cells. I'm pipetting all of this media off. So now the dish is empty. I'm going to take this tip that has been in touch with cells and place it into biohazard waste, which is below me right here. I'm going to put that away for a second. And now what I need to do is I need to rinse these cells. And to rinse the cells, I'm going to use 5 mils of DMEM. So remember, that's media without any FBS. The way we do this is we're going to use the automatic pipetter and a plastic pipette that's individually wrapped and sterile. So I've only opened the top, and I'm going to put the pipette, I'm going to seat it on the pipetter, and then I'm going to make sure that my DMEM is open and available to me. And now I'm going to take this cover off, exposing that. Open my DMEM by picking up the purple lid in my 2 first fingers. You push the top button and it sucks up the media. Now I'm going to place the media, the DMEM, in the dish by pushing the second button. And I don't want to spray directly at the cells. I just want to go down the edge of the dish and then kind of rinse them a little bit by moving them back and forth. You don't want to shake them really hard because they're like little people. You don't want to shake them up too much. Okay. This pipette goes into the biohazard waste bag. that is in this container, because this is a much thicker bag, and so it won't poke anybody when you're trying to throw it away. Now, we've rinsed our cells, so now once again we need to aspirate out that rinsate, which is, remember, D-MEM without any FBS. Fingers underneath, use my thumb and forefinger to open it. Aspirate out the media. Throw the tip away. And now we want to get 1 ml of trypsin, and the trypsin is an enzyme that's going to break the protein interactions with the bottom of the plate, and so that's going to cause the cells to release. So I'm going to move my trypsin to the front, loosen the top a little bit. I've already attached my pipette. I've got a 5 ml pipette, the automatic pipetter. You never want to touch the tip of this because that's what's going to come in contact with your cells. So I'm going to get 1 ml of trypsin. And I'm going to add it to the plate. Dispose of that pipette. Then what you want to do is you want to make sure that the trypsin covers all of the cells. Now you can take this plate and you can put it back into the incubator for no more than 5 minutes, or you can just let it sit here on the counter, and while that trypsin is working to get those cells to break off. While that's happening, what you need to do is set your dishes up for actually doing your split. So we're going to split our dish of cells into 2 populations of cells. We're going to do a 1:4 dilution. Okay? And to do a 1:4 dilution, what we're going to do, it's kind of counterintuitive, but we're trying to decrease the number of cells that we have in this plate into these plates. So I'm going to add 4 mils of media to this once I take the trypsin out. And I'm going to add 9 mils to each one of these new plates. Before I can add anything to anything, I need to write on my plate. I need to tell the cell type, which is 293 T cells, the passage number, which is pass number 6, because this one was pass number 5, my initials, and the date. I'm going to do that on both of these plates. So to get ready for my dilution, what I'm going to do is I'm going to add 9 mils of complete media, so that's D-MEM, C-D-MEM. So I need a 10 ml pipette. Have that, I have my pipetter. And the other thing I need is I need some C-D-MEM. And rather than take directly out of this large stock bottle, what I want you guys to do is, if there's not a small tube of C-D-MEM in the refrigerator already, I want you to make one. So this is a sterile 50 ml tube. I'm going to loosen the top on it. And I'm going to loosen the top on the stock bottle. So this is our C-D-MEM. And I'm going to pour some of our C-D-MEM into this smaller tube. So to do that, I'm going to take the top off my C-D-MEM, take the top off my clean, sterile tube, and I'm just going to pour from the stock bottle into the 50 ml tube. Cover my stock. That way it's not exposed to the air too long. Okay? Now this is C-D-MEM, so I want to make sure and write on this. I want to write the date and my initials because I'm the one that did this. Now what I want to do is I want to take 9 mils of complete media because we're getting ready to put cells in there. I want to make sure that my CD mem is open so that I can get to it. CD mem, I'm going to put 9 mils in each plate. Now I can use the same pipette here because I haven't touched anything that's not sterile. Alright, so the next thing I need to do, because my cells have been sitting here for a few minutes, I need to get some complete media on these so that we can shut the trypsin down, because the FBS has enzymes that actually break down the trypsin. So I'm going to add 4 mils of media to my population of cells that I've got here on my confluent plate. Now, I want to mix this up really well, and then I'm going to rinse the cells off the bottom of the plate by pipetting up the media, trying to be careful not to get too many bubbles, and then kind of shoot it out and rinse the cells down. I'm going to do this about 3 or 4 times. Now what I've done is I've pipetted up most of the cells, and for my 1:4 dilution I'm only going to take 1 mil out of this 4 mils that I've added and add it to our next plate. So I'm adding 1 mil of cells here and 1 mil of cells here. I'm going to return these cells to this dish, and we're going to throw those away in just a few minutes. Remember, into the sharps container. And so we have 2 populations of cells, and this is going to be a 1:4 dilution, which I'm going to write on here, because what we've done is we've taken our population of cells that was in our plate, we've added 4 mils of media to that, and we've taken 1 mil of those cells and we've put them into each one of these plates. So that's a 1:4 dilution. Okay? Now, these plates are ready to go into the incubator, so I would take them right over to the incubator and put them in there. Before I do that right now, what I'm going to do is I'm going to get rid of these cells by getting a little bit of Clorox, sodium hypochlorite, and actually spraying it directly into the dish. And when I do this, there's a change in color to purple, and eventually it actually goes to a clear color. I want to get rid of these cells, and I want to get rid of this tip, this pipette. So I'm just going to use it to vacuum up or aspirate up this wash media here, Clorox. Okay, now this pipette needs to be put into the sharps, and this dish goes under the hood into the biohazard media there. And now I'm going to take my cells and put them in the incubator. The last thing we need to do is we need to make sure and take the hood down, so we want to make sure to turn everything off and put everything away and clean it so that the next person that comes in, everything's ready for them. So just want to move everything to the back and get rid of that. And okay, we're going to be putting all of the things that we got out of the refrigerator away. So any tubes that we want to put in the refrigerator, we want to make sure the tops are on very well, that they're very clearly labeled, and that we have put parafilm on them to make sure that nothing can get into them. So we have little pieces of parafilm in the hood. So you just want to put that around the edge of each one of the tubes. these away in the refrigerator. Plus, you want to make sure that you put, thank you, you want to make sure that you put parafilm around your stock media. Just one more barrier to help with minimizing contamination. So these are all going to go back into the hood, into the refrigerator. I'm going to put my pipetter on the stand, throw away any trash that I've got. And I'm going to turn the light out real quickly. Now, we can see the flame here. Before I actually do the cleaning in the hood, I'm going to turn the flame off. Okay, so that means to turn the gas off. Okay. And now, if I spray ethanol in the hood or anything, it's not going to blow up. So, but I can spray ethanol, wipe it up really well. And it doesn't hurt to give it a little squirt of Clorox. So we would re-plug in our pipetter before we leave and turn off the aspirator and put everything back into the refrigerator, and you make sure and turn off the fan.